Good afternoon. Thank you for coming here. I am Sung Kim and the CEO of OncoBix. Today I'm going to talk about the anti-cancer drug discovery platform of OncoBix and then its application. But unfortunately, today's my talk a little bit deeper from this session's topic, the bispecific antibody. But however, I hope my talk will give you another perspective and possibility of another antibody drug application. So before start, let me introduce myself and my company. I graduated from Department of Chemistry at Gong University and graduated from also the College of Pharmacy, Seoul National University. And then I moved to the United States, and I have worked at the National Cancer Institute, NIH, in the United States as a postdoc for 5 years. And then I moved back to Korea, and then I started my own business 2016 based on those experiences. So since then, my research was focused on the kinase inhibitor development, the discovery and the development. And then I also expanded my the network and then collaboration to the industry, academia, and the institute. Now we have the good relationship and the partnership with our many collaborators, such as JUTH Biotech and Asan Hospital, and then Gyeonggi Biotech, and then the Pasteur Institute. This slide shows about the drug discovery process, as you've seen before many times, but I can explain it in shortly. As you know, to create a new drug, it needs a study for evaluating efficacy and the safety. Clinical studies is a process to compare whether the drug candidate are beneficial to the human, following the research processes such as basic research and the non-clinical studies. Clinical studies are ordinarily divided into 3 phases, and they performed at the medical institutes such as hospital under very strict regulation for healthy volunteer and the patient. So recently, the antibody drug conjugate have been drawn attention. So in shortly, I said the ADCs. ADCs are a class of biopharmaceutical drug designed as targeted therapy for the treating cancer. So unlike chemotherapy, ADCs are intended to target and kill tumor cell while sparing the healthy cell. So that means with better potency and then safety and then less toxicity. So ADCs are as of 2019, about 65 or 6 pharmaceutical companies were developing the ADCs. So ADCs are complex molecule composed of antibody linked to biological active cytotoxic payload, like natural toxin and artificial toxin. So because using the toxin, sometimes ADCs raise the safety issues. So as you see in previous slide, part A shows a simplified general structure of ADCs. Part B shows the general mechanism of the action, the ADCs. So let me explain it shortly. Step 1, the ADC binds its target cell surface antigen receptor to form the ADC antigen the complex. And then step 2, leading to the endocytosis of the complex. The step 3, the internalized complex undergoes the lysosomal processing, and step 4, the cytotoxic payload released inside the cell. Step 5, the released payload binds into its target, and then step 6, leading to the cell death. So now more than 50 different known antigens have been used as a target for ADCs that currently in clinical or clinical development. A few years ago, the Miao group in Ningxia University in China suggests that compared with the traditional ADCs, small molecule drug conjugate, in shortly SMDCs, provide a new point of view, but less established perspective for targeted therapy. In spite of this, SMDCs have several strong points. First, they have non-immunogenic properties. Second, they match more the manageable synthesis, much more than the traditional ADCs. Finally, the low molecular weight with a higher potential for cell penetration, even in solid tumors. So, SMDCs might be a promising alternative with similar efficacy to ADCs to increase safety and decrease toxicity. So, now we have some question. Which is more contributes in the SMDCs component? Of course, SMDCs use their appropriate the combination. So among the SMDC component, how can it discover new small molecule cytotoxic payload as a better potency and the selectivity and the safety, the less the toxicity? So at this point, we need some strategies. On this image, along the x-axis, we have two options: the known protein structures and unknown protein structures. The y-axis are the options. The y-axis are known ligand structures and unknown ligand structures. So depending on the combination of these options, we may follow the different drug discovery strategies. The most beneficial is a blue zone. It shows that what can be done when both protein and ligand structures are known. The green zone is next best. When the protein structure is known, but ligand structure is unknown, and so forth. The structure always will accelerate the drug discovery process. So our OncoBix is proposing the small molecule payload discovery strategy. Our platform technology, target-oriented focused for, in shortly, TOPAP, is originates from fragment-based drug design combined with the computer-aided drug designs. Actually, TOPAP has been created using green zone drug discovery strategies. The basically fragments are small molecule of less than 300 Dalton in size, contrast the larger and the more complex molecule using the conventional high throughput screening. So we properly use the fragment for three main regions. At first, a few hundred fragments can cover the same chemical space, the hundreds of thousands of conventional compounds. Even in a small lab like us, making screening feasible. Second, the fragment can be efficiently measure the protein, the druggable, the pocket, shown in here, the yellow circle. Finally, the fragment can be readily optimized by growing, the merging, and linking of the multiple hits, whereas optimization of conventional compounds is often very challenging. Instead of using the conventional expensive time-consuming tools like SPR, NMR, isosomal titration calorimetry, we are using the computer modeling. Computer modeling, the computer modeling is easily accessible while saving time and resources. Also, computer modeling makes us easier to understand the binding pattern between fragment and protein. After optimization, hit and read compound, we can propose the effective cytotoxic drug candidate after insights, chemical synthesis, and efficacy evaluation from our collaborator. So this illustration shows a SMDC structure with the applied top-up derived cytotoxic drug in briefly. As a result of applied TopUp, the platform to find the drug candidate in 2018, we could suggest an anti-cancer candidate named OBX1 and published the target oncology in International Journal and applied a patent. OBX1 compound identified the highly effective mutant selective EGFR TKI for the treatment of T790M mediated lesions in non-small cell lung cancer. On the left side is a simple flowchart of TOPA process, and then right side is a biological data by evaluating inhibitory capability at the cell-based assay in the published paper. The result table on the bottom side, compared with the other mutant selective EGFR TKI, such as almatinib and osimertinib, the OBX compound is a better potency and the selectivity for mutant EGFRs. So OBX112 compound treatment was very highly effective against the human EGFR mutant lung cancer models with or without EGFR T790M, not only in vivo or in vitro, but in vivo. So for example, if we combine this result, we can design SMDCs applied our cytotoxic candidate OBX112 to increase the safety and then decrease the toxicity. Of course, I understand that to develop that drug, we still have a lot of barriers. But I think in spite of this, SMDCs might be promising alternatives with a similar efficacy to ADCs. To summarize, the novel classes of small molecules have been developed as a tool for treatment of cancer and other diseases through the different approaches like ADCs, immunotherapy, and then protein degradation. Within the growing the research area, progresses in the ligand screening and the procedure and the hit-to-read optimization. Our platform TopUp technology helps to find the cytotoxic drug discovery easier to increase the safety and then decrease the toxicity, and combined with the advances in the various drug release technology, using the antibody. So now we're making the way the future treatment of the broad range of pathologies with the targeted therapy. The future is right now. So well, that's all I have to say. Thank you for your kind attention.